Hello and welcome everyone to the first in a series of informational webinars exploring MediciNova's pipeline of treatments in clinical development for difficult to treat diseases and conditions. Today, we will discuss a very important topic, ALS, and MediciNova's clinical trial, Combat ALS, with two esteemed experts in the ALS medical community. Dr. Bjorn Oscarsson, the lead principal investigator of the Combat ALS clinical trial, and Dr. Benjamin Brooks, who led the first clinical study to evaluate MN166 in people living with ALS. First of all, Dr. Oscarsson will provide an overview of MN166 and the clinical trial, Combat ALS. And then Dr. Brooks will discuss important findings from the phase 1b/2a trial that laid the foundation for the Combat ALS study design. On June 21st, the International Alliance of ALS MND Associations recognized Global ALS MND Awareness Day. MediciNova joins other organizations and foundations to raise ALS awareness all over the globe and to find safe and effective treatments. We are committed to these common goals. Although most people living with ALS have similar symptoms, it is not a uniform disease. Each person exhibits different symptoms, experiences different disease progression rates, and reacts differently to treatment. For example, Dr. Stephen Hawking lived with ALS for 55 years. Ms. Saeki, a beloved Japanese TV celebrity, on the other hand, lived just 9 months from the time that her first symptom appeared. Now, Dr. Hawking and Ms. Saeki's cases are not typical, but they underscore the complexity of this disease. Now let's welcome our special guest to the webinar. It is my pleasure to introduce Dr. Bjorn Oscarsson, lead principal investigator of the Combat ALS clinical trial conducted at more than 20 sites across the US and Canada. Dr. Oscarsson is a neurologist and neuromuscular disease specialist at Mayo Clinic in Jacksonville. Dr. Oscarsson will provide an overview of MN166, including its history, mechanism of action, and its neuroprotective effects. He will also describe the Combat ALS study objectives, design, eligibility, and phases. Dr. Oscarsson, thank you for being here.
Thank you for having me and letting me talk about this exciting project. So a little bit about MN166. The way we're studying it here is in a easy to take small capsule, which is quite small and relatively easy to take even if you have swallowing problems. This is a drug which has been used in a somewhat different form for a longer time in Japan. We have about 20 years of experience with the drug there, with a large number of people having been exposed to the drug. And there the experience has been favorable as far as the safety of the drug. This dose that we're studying in ALS is a bit higher, but even at this higher dose, the safety experience has been quite good. There has been some side effects reported, but really not very much. The study that you'll hear about from Dr. Brooks that really has laid the foundation for the current Combat ALS study did show some interesting and thought-provoking findings. There's also one study in progressive MS that we'll briefly touch on, and in both these there were positive outcomes that we were hinted at least. There were more responders in the ALS function rating scale, meaning people holding stable levels or maybe even slightly improving over time, as well as with the similar measures ALSA-Q5 and for muscle strength. And in the SPRINT study, we saw that the brain shrinkage that is associated with MS was slowed down. Next slide, please. The effect of MN166 in neurodegenerative diseases is probably several-fold. So it may not be a single effect that is responsible for these tantalizing findings. There may be a set of different things. There's many of them that are thought to be related with neuroinflammation and the activity of the inflammatory cells in the brain. And again, that's an effect on the macrophage migration factor, phosphodiesterase inhibition, and toll-like receptor 4 receptor inhibition. So on the other hand, there's also some evidence suggesting that the drug has an effect on how proteins are broken down in the cell. We see that there is problems in many neurodegenerative diseases with how larger protein complexes are broken down. So sort of leftover garbage proteins that are accumulating in the cells, and we think that that is part of many neurodegenerative diseases. Next slide, please. So much of the original work on the drug has focused on inflammation and neurotrophism. So these are different substances that are changed in a positive direction, in a favorable way, suggesting that the drug may be of use for neuroprotection. And next slide. This is a list of some of these studies that have support the notion of that MN166 or ibutylast may be useful for neuroprotection. We have the top study there does speak about glutamate-induced excitotoxicity, which is one of the factors that we think are important in ALS. It's the aspect of the disease that we think that we are predominantly affecting when we're using the riluzole drug. But again, here, most of these other studies talk about different inflammatory aspects of the drug, or anti-inflammatory aspects of the drug. And next slide. Here, there have been both a study in Japan of relapsing remitting MS, where there was a suggestion of that the drug slowed the loss of brain volume loss of the protected brain. And that was repeated in the SPRINT study, which is the phase 2 trial of ibutylast that's on the slides here. This study did show that on MRI imaging that there was less loss of brain volume. Again, we think more of multiple sclerosis as a neuroinflammatory disorder than a neurodegenerative disease. But it is a disease that probably has both facets, which again, maybe in ALS, the neurodegenerative aspect will be dominating and the neuroinflammatory secondary, and MS might be more the other way, but these are intertwined concepts. These types of experiences have suggested that the drug can be used in these kinds of diseases with reasonable safety. And I'd say next slide. This study, which is more recent, highlights the idea of that the drug can be important in removing misfolded and abnormally aggregated proteins in ALS models. The study here showed that TDP43, which is the type of protein that we see in most forms of ALS, was able to clear better with the addition of the MN166. This was also true for the main alternative protein aggregate, the SOD1 protein. So this evidence does can speak towards that there could be yet another way that MN166 helps protect motor nerve cells in ALS. And next slide. So we have an incomplete understanding of the many processes that go wrong in motor nerve cells in ALS. This is a top 10 list of things that we know are going wrong, but it doesn't really speak to the core issue, sort of what the first problem is in ALS. And we already today have drugs that work on supporting mitochondrial function and excitotoxicity. But it's entirely possible that since we're kind of approaching the disease at the later stage, that we will need to be hitting the disease in many different places to really achieve a meaningful effect that adds up to helping patients. And this drug could have that potential. We are seeing that MN166 does engage on multiple fronts here. In the order that the processes are listed on this slide, again, MN166 does address this process of proteostasis, or breaking down these protein aggregates. So that is one thing where we could see an effect. Next one. And we're also seeing, again, the neuroinflammation and attenuation of the microglia. That this is the more classical thinking about how this drug works. And we can see an effect there. And lastly, there again is evidence that it ends up having some ability to reduce excitotoxicity. So these are all ways where we possibly could end up with a sort of multi-hit on the disease process. Next slide. So the study that we are now doing to really see if the data that Dr. Brooks had given can hold true and give us sort of flush that out to see if there really truly is the effect that we're hoping for with this drug, is the Combat ALS study. This is a longer study. It's a 20-month study that we allow for a 1-month screening period where patients are going through this. This can go quicker. Again, want to make sure that people, again, are the right candidates, that there's no safety issues for people enrolling. Then we enter into the double-blinded treatment phase. This is it's a year-long study where we're looking at either active drug or placebo. We do think that this type of placebo study with an allocation of 1-to-1 gives us the greatest power to be able to see an effect of the drug. We are always looking for ways to try to minimize placebo exposure, but to have the smallest number of people in a study, a 1-to-1 allocation is the best, and we have settled for that here. This drug is taken twice a day and escalating doses over the study. We have some flexibility in that lets us keep patients in the study, I think, with some flexibility. After that, everybody who enters the study and completes the year is eligible for an open label extension, where you as a volunteer are guaranteed to be getting the Ibutalas drug. And that lasts for at least another 6 months. And then we have additional follow-up after that. The study does have a smattering of clinic visits, as you can see on the bottom. They are relatively infrequent, consistent with most studies at this stage of development. Next slide, please. So again, it's screening baseline. Then at week 6, you come in, again, month 3, 6, and 9, and the at 1 year is the combined end of the double lump phase and starting of the open label extension, the OLE phase, where we then follow out for another 6 months. Next slide. The key inclusion criteria or eligibility criteria is age 18 to 80, having ALS, the disease duration, so that from time of first weakness to getting into the study is short. So we have selected this to try to make the study answer the question of if the drug really works or not. And again, there are statistical reasons why this time point has been selected. And looking at other studies that have been done in the last 5 years, this is similar to the time points that were selected for the Mitsubishi study 19 and Amylyx's Centaur study, which were able to show an effect. So that is something which we're definitely also mindful of. Again, we do want to pick up on the effect here, again, if it's really there. We do want a relatively high level of function at entry with both on the ALS-FRS, the ALS Functional Rating Scale, and also near normal breathing functions, again, 70% or better. These capsules, again, are small and relatively easy to study, but yes, people do need to be able to swallow the study capsules to be able to enter into the study. The dosage escalation schedule is there to your right. So again, we start a bit lower and then increase. Next slide. The main endpoint, what we're hoping to see in the study, is that the drug has an effect on the combination of survival and loss of function. And so we'll be using that as a combined endpoint to see if the drug can affect that. And we're also looking at these independently, both the change and the survival. We're going to be looking at a responder analysis. That is a type of analysis that many people, it makes sense, I think, from a patient perspective. And if you're able to, even if a subset of people are able to hold steady or in their disease, that will be very valuable. And so we will see, again, if we can see such a difference. In this study also, that was something that Dr. Brooks was able to show earlier. Certainly safety and tolerability are still things that we want to look at in this larger ALS population with this higher dose and longer time. And we want to see how the blood levels of the drug affects these things also. The next slide. The study right now have 19 sites in North America. So they're hopefully a site not too far from you if you are interested. Next slide. And I will be handing over to Dr. Brooks, so maybe by way of Katty.
Thank you so much, Dr. Oscarson, for that presentation. Now it's my pleasure to introduce Dr. Benjamin Brooks, Professor of Neurology at University of North Carolina. Dr. Brooks, a sponsor investigator, led a single-site phase 1b/2a study that evaluated MN166 for the first time in people living with ALS. The findings from his clinical trial laid the foundation for the Combat ALS study. We are so pleased to have you here, Dr. Brooks.
Thank you for this opportunity to discuss this important issue. Next slide, please. There were positive findings from the initial phase 2 study with respect to adding ibudilast to riluzole in treatment effects. There was the excitement from the NIH-sponsored NEXT trial of ibudilast in multiple sclerosis was one of the reasons we pushed forward with this trial in amyotrophic lateral sclerosis. The ibudilast trial in multiple sclerosis showed decreased brain atrophy with treatment. And brain atrophy is one of the features that occur in ALS as well. Next slide, please. The ALS 1201 study was a placebo-controlled randomized double-blind study for 6 months, followed by an open-label extension phase. Patients were eligible if they had onset of ALS within 5 years, based on the original Riluzole criteria. Patients were randomized 2 to active treatment and 1 to placebo. The total number of patients started were 51, 17 on placebo, and 34 on treatment. The safety population was 51, all intention to treat. The full analysis set was 49 patients. All of those patients achieved 1 post-treatment evaluation. Next slide, please. The responder analysis was based on looking at patients who had stable or improved score at month 6 at the end of the double-blind phase. And this was shown in terms of the treatment with respect to the ALS functional rating scale total score, where 2 patients were improved on the placebo and 7 patients were improved on the treatment phase. The ALS quality of life scale showed 4 patients improved or stayed the same, and 17 patients in treatment were improved or showed the same. Manual muscle testing showed that 4 patients on the placebo side and more than twice that on the treatment side were noted. Because of this analysis, we felt that we should look more precisely at what was the benefit of this improvement. Next slide, please. The responder analysis on the treatment side, placed on the left, and the placebo side, placed on the right, shows that more patients were stable during the treatment phase of the double-blind phase than in the control. Next slide, please. We know from previous studies that if there is slow progression, then there may be better survival. So we looked at the survival of all patients who participated in the clinical trial during the double-blind and open-label extension phase and followed all of these patients post-treatment. As you can see in the blue line, those patients who were not able to complete the protocol had a median survival of approximately 24 months. Whereas in the red line, those patients who completed the full protocol had a median survival of 36 months. Now, this suggests that if you can delay the rate of progression of ALS, you might have a benefit on survival. This has been shown recently in other clinical trials as well. And this is one of the main reasons for proceeding with combat ALS. Next slide. This is a statistical analysis showing that the benefit was statistically significant in terms of starting treatment early and having benefit and able to continue treatment with ibudilast and Riluzole for the full treatment period. Next slide. Subjects who completed 6 or 12 months of treatment showed improved survival at 30 months post the initiation of treatment, even though they had stopped treatment at the end of 12 months. Next slide, please. Further analysis showed that those patients who had bulbar and upper limb onset may have shown the dramatic effect of that benefit. So we analyzed the patients in those different categories as a function of disease onset, and also noted that there was a difference in terms of the number of patients who were earlier in the course of the disease that showed improvement compared to those patients later in the course of the disease who did not show improvement. For that reason, the protocol was changed to limit the patient to within 18 months of onset, as Dr. Ossikin pointed out. Next slide. There's a wide variability in the rate of progression of ALS, as shown in this slide. Item A shows all of the patients, but these can be broken down into those who have rapid progression, like in panel C and D and E, and slower progression in F. This has been confirmed also by what is called the Answer ALS analysis and also by the PROACT analysis. So the goal is to see whether we can slow the rate of progression in the early phases of the disease in all those patients receiving treatment in the first 12 months compared to placebo. Next slide, please.
Dr. Brooks, thank you very much for sharing the encouraging results from the ALS 1201 trial with us. You have both given us valuable information and hope about MN166 and the ALS trials. Dr. Oscarson, would you mind revisiting the highlights of the Combat ALS trial for our audience?
I would be happy to. So again, this study that we now are doing to kind of show if this can hold true. Again, we are very excited about this evidence that built us, that gotten us this far. We have the short duration of disease, again, that is built both on statistical reasons, experience from other studies, and the data from Dr. Brooks. We do want people not only with a short disease duration, but also a high level of function coming in. We calculate that rate of progression leading into the study to make sure that that we're not selecting people that have too slow disease to be able to inform us of if the drug works over this year duration of blinded study. We are wanting breathing functions at the entry into the study to determine if how advanced people are in that aspect of their disease, and make some guess as to that we are going to be able to continue to work with people that they are going to be able to dose the drug for some time. But we don't necessarily require these breathing functions to be done right at as part of the study. We can utilize previously obtained breathing functions that are relatively recent. We aren't primarily looking at the breathing functions as the primary outcome measure. So that gives us some flexibility in how we're using the breathing functions during these times of COVID, where breathing functions have become more complex to do. We can even let participants do their breathing functions at home if that is necessary to kind of get people enrolled in the study. Again, the study does have an open label extension phase, which is a good feature. It adds to our safety data and guarantees that everybody who makes it through that year-long study is going to be able to try the MN166 Ibudilast, at least for some time there.
Great. Thank you, Dr. Oscarson. Despite the rarity of this disease, its impact, as most of you know, on people living with ALS and their families cannot be overstated. This is one reason why MedAct was established, to lessen some of the burdens, such as, for example, long distance travel, which could be a huge barrier to otherwise eligible patients' enrollment in the clinical trials. MediciNova recognizes that traveling to a faraway clinical site is not easy for a person living with ALS. Even though there are about 20 sites across the US and Canada, for now that doesn't accommodate every potential participant, and we want to remove that hurdle. For more information on Combat ALS trial, please visit clinicaltrials.gov and search for the Combat ALS trial under this NCT number, or contact us via email at inquiries@medicinova.com. MediciNova is a biopharmaceutical company whose goal is to find safe and effective therapies for diseases and conditions with strong unmet medical needs. MediciNova is committed to upholding the highest standards of integrity, safety, and quality in the conduct of their clinical trials. Clinical site investigators and staff participating in the Combat ALS trial reflect our philosophy. Thank you, Doctors Oscarson and Brooks, for sharing your findings with us, and we look forward to hearing your updates about the Combat ALS trial. And a special thank you to you, our audience, who are watching this webinar. Please spread the word about the Combat ALS trial with your family and friends, and especially within the ALS community. We'll see you next time.